Good afternoon. Thank you for the invitation to present at the 6th Annual Congress on Plant Science and Biosecurity. My name is Chris Matocha from the University of Kentucky, Martin Gatton College of Ag, Food, and the Environment. And today I will talk about some work dealing with transformation of organic matter under anoxic conditions in soils. The storage of soil organic matter is the net effect of the additions of carbon inputs through plant net primary production at the surface and the loss of carbon through decomposition. Here's an example of a soil pit, the Sadler silt loam, one of the soils I use in this study, an oxyaquic Brach-Glass-Udalf, and you will note the darker area here at the surface corresponding to about a 1.2% soil organic carbon and total soil nitrogen of about tenfold less. And so this accumulation of carbon is desirable from the standpoint of carbon sequestration, as well as all sorts of other important properties in soil, such as water holding capacity, availability of nutrients, and so on and so forth. So regarding decomposition, it can vary depending on whether oxic or anoxic conditions prevail in a given soil. For the transformation of organic carbon via oxic transformations, in other words, where oxygen is present, the prevailing view is that large biomolecules representing organic matter in a soil undergo oxidative depolymerization to form monomers, smaller compounds, which can then be utilized by microorganisms under aerobic respiration where oxygen is present, producing then CO2 and energy. Now, when all the oxygen is lost, which could be a situation, particularly in the Sadler, where you notice these interesting color changes. It has a fragipan, there are grays and mottling going on. It is a moderately well-drained soil, and you notice oxyaquic. It can be flooded, particularly in the springtime. And when that happens, oxygen can be lost, and hence anoxic carbon transformations can then occur. And the model here, the paradigm is that large biomolecules undergo hydrolysis to monomers. But what then occurs is these monomers then undergo fermentation processes, producing low molecular weight substrates, fermentation products such as acetic acid here, CH3COH, and hydrogen would be another example of one. These fermentation products react with native terminal electron acceptors in a process called anaerobic respiration to produce then CO2. All right. Now, so these terminal electron accepting processes, after oxygen is depleted on the redox ladder, next we have nitrate going to dinitrogen in a process called denitrification. Or it could then be manganese 3 and 4 oxide going to manganese 2, and then iron 3 going to iron 2, sulfate going to disulfide. And so these fermentation products can be utilized in this sort of scenario. And the prevailing view I have listed in red is that fermentation is slow, meaning it's rate determining. And things like acetate don't accumulate to any significant extent because once they're produced, it's slow. They're immediately utilized by respiration processes. So this is rapid. And this is work done in the biogeochemistry literature, including Diek Posma, who's done some of the classic work looking at this process and providing some nice guidance here. So that's the prevailing view. These TEAs, or terminal electron accepting processes, occur at different redox potentials, and that's indicated here in this particular plot where I have redox potential normalized to pH 7. And as we go from your right to left, we go from an oxic situation where oxygen being reduced to water occurs at a measured value here, a little bit less than 400, 350 to 400, followed by nitrate next on the list, or next on the redox ladder, then manganese 4 going to manganese 2. You begin anoxic conditions where iron 3 going to iron 2, followed by sulfate to sulfide, and then lastly methanogenesis, where carbon then is producing or is being converted to methane. Okay, so these occur at these various redox potentials uh, in this redox ladder. So, paradigm for anoxic carbon decomposition: fermentation slow, anaerobic respiration is rapid. Okay. Um, and at the same time, what we wanted to look at in this project, because there did seem to be a good handle on it, is how does ammonium production under anoxic conditions fit in to fermentation and respiration? Some early work on this to try to understand production of nitrogen and its availability to plants. I actually found that anoxic carbon production of ammonification, was a good indicator of plant available nitrogen to plants over a growing season. So we wanted to look and see how that fit into these other processes, sort of testing the hypothesis, if you will, that um, fermentation is slow, anaerobic respiration is rapid, and then seeing where ammonification fit in. And so the objective of the study then was to investigate fermentation, respiration, and ammonification in stirred batch microcosms under anoxic conditions by measuring chemical species over time, using native terminal electron acceptors and soil organic matter in four soils varying in drainage status. Now, the four soils we collected were from the western part of Kentucky, 0 to 10 centimeters, and we collected these from native grassland sites. The Sadler, which is a moderately well-drained soil, sort of on the gentle side slope of in the landscape position, and we collected that here just south of Princeton. Now, the other three soils we used, the Feliciana, Granada, the Callaway, were all part of a catena, with the Feliciana being the well-drained end member at the summit position, Granada at the gentle side slope, and the Callaway here at the back slope position. Poorly drained, moderately well-drained for the Granada, and well-drained for the Feliciana. So 4 different soil types we utilized. We then took these soils back to an anaerobic Coy chamber, where we then prepared soil microcosms, essentially slurries. And then, as I said before, using native terminal electron acceptors and native carbon. We did use an argon atmosphere, argon only. We did not include what these coil chambers recommend, which is a hydrogen gas to remove all the oxygen, just because, as we'll talk about later, hydrogen can be juicy for microorganisms. So we had argon only, and then began to follow these things. Here in blue, here are the things we followed. Nitrate concentrations, manganese 2 concentrations, iron 2 concentrations, sulfate levels, and ammonium. Sulfate, nitrate were performed using ion chromatography, and then the others were colorimetric procedures. And we also measured soil EH and pH using separate aliquots to try to better understand what's going on. So here is a table of the properties of the soils, the 4 soils, the well-drained Feliciana. These first 3 are from the catena. A couple points to be made. Number one, as you get worse drainage in general, carbon tends to increase, which is what we find in textbooks, because aerobic respiration in general is thought to be much faster as well, CO2 production there. And so you blow off carbon more so than you do in an anoxic situation, okay? What else of note? These other 2 soils, the Granada and the Callaway, actually had very high levels of native nitrate compared to the other soils. The Sadler, the moderately well-drained, and I should have pointed out this was in a different location, not part of the catena, and also is formed in a thin mantle of loess, which is underlain by sandstone, I should point out. And you see the sand content more than double what the other soils have, and a little bit higher pH and CEC that's modest compared to the other guys. All right. So now we're going to be following some of these microcosm data. Here on the y-axis, we have ammonium, manganese, iron, and acetate, okay, on this axis. And on this axis, we have sulfate or nitrate. And this will be similar for all the next plots I show. And this is for the Sadler, the moderate drainage soil. And then I'll show EH and pH here shortly. So a couple important points to make. You notice there's a slight increase in sulfate if we just go from the top and bottom. Sulfate, slight increase. And then we see a drop in sulfate, okay? Now, what happens with other things? Acetate here in the open symbols tends to increase, maybe reaching a parent plateau, and then it kicks on again. at longer times. Ammonium and closed symbols tends to co-occur, you notice, with manganese 2. Okay, manganese 2 and ammonium, very similar patterns. Iron, there's a bit of a delay before iron begins to be produced. You kind of see that throughout. And I'm going to show rates. What we did for rates was we looked at the initial first few linear data points, as I'll show here in a table in a little bit. But important point to make, acetate comes out immediately, and it co-occurs with manganese 2, and then after 4 days or so, then we start seeing iron 2. Okay. I should also point out there's very little nitrate in this soil, as I saw previously as well with the characterization data. Now, the EH and pH for this soil, moderate drainage soil, in general, EH starts off at about 100-ish and tends to decrease pretty quickly down to a very low negative value of about -80, and then doesn't change after that. Starts off about 100 millivolts down to -80. pH starts off about 7.1, and it kind of tends to increase over time. Now, Feliciana, this is a well-drained soil. How does it compare to some of these others? Well, first of all, it does have a little bit of native nitrate in there, and then the nitrate drops immediately. We see something similar with sulfate, where it tends to be higher sulfate values here, and then they tend to drop over time. What else happens here? Well, I see ammonium and manganese tend to increase here after a few days. Acetate doesn't kick on until about day 14. Then it kicks on, okay, acetate values, and then it tends to increase just as the manganese and the iron do. Iron then really jumps up here after about 7 days and then 14 days. Notice in the green symbols for the Feliciana. Now, what are the redox potentials doing? The Feliciana, being a well-drained soil, tends to be higher the very first data point after 24 hours compared to the Sadler, and it drops. See a sort of exponential decrease here, and pH tends to be sort of stabilized in the Feliciana. Now the Granada, which is the moderately well-drained general side slope position, just down from the Feliciana, notice a lot of nitrate here. Okay, if we just follow these data, nitrate tends to be high, then it decreases over time. Sulfate tends to just increase over time. And what do we have here? We have, in general, we have manganese tends to kick on, okay? After about 7 days, we start seeing manganese. Iron never increases, so we never reach iron-reducing conditions, but we do reach manganese-reducing conditions. And acetate value is very low, slight increase. It's hard to tell with the scale, but just ever so slight increase here in the Granada. Now, what happens here in terms of redox with the Granada? Well, the Granada is very oxidizing compared to the well-drained Feliciana. Notice here, EH values tend to be 400-ish, slight decrease, okay, slight decrease over time. pH tends to go up a little bit, okay, but this was a little surprising to us, and you might recall the higher nitrate levels, and I think that's what's going on, is that native nitrate tends to be poisoning this redox high, poisoning it to higher redox values, and then it does tend to decrease over time as we see a loss in nitrate. Now, the last soil in the lot, the poorly drained backslope soil, the Callaway, much greater, and same data, we look at much greater nitrate concentrations here as compared to the Feliciana for sure, and a little bit more than the Granada, but nitrate decreases immediately, okay? What else happens here? Sulfate, we never reach sulfate reducing conditions. Ammonium tends to have a moderate increase, and then we really don't see much. Well, we see a lot of manganese kicking on here. Manganese tends to increase here in red, particularly day 5, day 7, and then following. And then iron tends to increase here as well. Okay. Now, what happens to EH and pH? These were corrected to a pH of 7. Because the pH started off very acidic. This is the most acidic soil in the lot, the Callaway. It tends to increase over time. And then look at the EH. It was even more oxidizing than the Granada. And this is the poorly drained soil, I might add, with the higher organic matter level. Higher EH, then it decreases over time, concurrent with some of the conditions we see with decreasing nitrate and all that. But I should point out, The most negative we get here is 0, and we never reach sulfate reducing conditions, but we do have manganese and iron reducing conditions achieved. Okay. All right. So let's compare these rates, see what's going on. And if you look at this table, we have soil in this column, drainage status, and we have all these chemical species that we measured. A couple important points to make. First of all, the acetate production rate in the Feliciana, well-drained soil, is no different than the manganese production rate in the HCl fraction. I didn't show that data, but we basically took acid extractable manganese and iron from the Feliciana and the Sadler. But it was no different. Notice similar symbols, 0.215, 0.119, 0.35, no different statistically, which means that the rates of manganese production, meaning manganese for respiration, and acetate production, in other words, fermentation, there's no difference between them, which would indicate that these processes co-occur, or they're decoupled. In the Sadler, we see something very similar. The manganese in the water fraction is a little bit higher here, but in terms of the HCl fraction, it's not. And so once again, co-occurrence, and we saw how the evolution of manganese and acetate were concurrent with one another as well. And so this was very interesting and surprising as well. Now, in terms of some of these other redox TEAs, for the most part, acetate was significantly less in the moderately well-drained Granada and the Callaway compared to when it came to other TEAs. Okay, what does that mean? That just means that respiration is much more rapid here than the Callaway and the Granada, which is which would then follow the model paradigm. Okay. Now, last point, ammonium production rates in this column. You notice the highest acetate production also corresponded with the highest ammonium production rate, which was in the Sadler. And then we kind of see that scale accordingly. So it's interesting. And so we then try to correlate these. Not that correlation means causation, but we correlate some of these with various soil properties. Okay. All right. So take home summary. Fermentation rates based on acetate production and manganese for respiration were similar in 2 of the 4 soils, indicating these are decoupled or they co-occur. This has been reported in several pretty recent studies in some marine sediments. So we might need to revisit this classical paradigm of anoxic carbon decomposition. Now, a nice study from Dennis Leroux at USC in Earth Science Reviews, he even reports studies in the marine literature where they find fermentation, there are some molecular biology techniques, they're finding that organisms that could ferment carbon under anoxic conditions were co-occurring with other species. That was kind of surprising. So he claims that, hey, maybe we've been thinking of fermentation all wrong here. So what are some reasons why acetate accumulated so quickly? Possibly preferential use of H2 or acetogenesis, which is where where acetate is actually produced. It's being produced in the process of CO2 combining with methane, actually. All right, and we didn't measure methane in our study. Fermentation rates weakly correlated with carbon and total soil nitrogen, and we kind of saw that. The soil with the soils that were the poorly drained soil, the Callaway had very little, had the most carbon, but yet a very, very slow fermentation rate. Now, sand content, R squared or R of 0.86, and dithion extractable aluminum were positively correlated with fermentation rates. So this speaks to maybe some mineral type interaction going on there. And lastly, I should point out initial nitrate levels were actually negatively related to fermentation. Now, a couple important points here. Ammonification rates and fermentation rates were positively correlated. What does this mean? Well, does this mean that amino acids were fermented? Could be. We didn't follow amino acid levels in the pore water, but this does speak to those of you who know this, the Strickland reaction, where amino acids can actually be produced. And what you could do is you produce ammonium and acetate in the process. Historically, it's been thought to play a minor role. And lastly, ammonification rates were positively correlated to manganese 2 production, which was shocking. We did not expect that either. And so we actually took sand-sized nodule from the Sadler and started characterizing it recently since that paper was published, trying to follow what's going on. And we see these honeycomb-looking minerals in this was basically a sectioned nodule that we opened up, we cut, and you notice here in red, we have higher values of manganese here than here. So this flake-like, cornflake-like, or honeycomb-like assembly of minerals were enriched in manganese, a little bit of iron, with a background of aluminum. When we take a EDS spectrum, you will notice that there looks to be maybe some 2-to-1s in the area or underneath, but this is definitely enriched 17 or so percent manganese with a little bit of calcium as well. This perhaps could be burnasite, could explain some of the trends with the sand correlation. Now, I'd like to acknowledge the student who collected all of these data, Stephanie Paizola, former master's student in my lab, who's now working for an aluminum company. This paper was in Stoughton back in 2025. And I'd like to acknowledge my collaborators, Jerry Mack, particularly with helping with the soils collection, and then USDA NEFA for funding. Thank you for your attention.